Hello, students. Welcome to Chapter 3 in our biopsychology class on the basics of neurochemistry. A lot about what happens with the neurotransmitters in the synapses, how do they get there, how are they created. Like a lot of the chapters, there's a lot of detail in these ones, so you don't need to have absolutely everything memorized, but some of the patterns, some of the major neurotransmitters that are brought up multiple times and focused on a little more, hopefully you're recognizing some of the commonalities between them of how they're synthesized, how they're used, things like that. And things like understanding a little bit more about like what is an amino acid and putting those together and those kinds of things. Some of the biochemical processes that make up what is going on in our brain. So let's jump into these slides. All right, you should still see my face there on the side or the corner. So here is chapter 3 on neurochemistry. To begin with, I want to point out these major categories of neurotransmitters. If you look in different places, in Wikipedia, for example, I'll show you their page in a few moments. But the book breaks up the neurotransmitters into a couple different key categories. One of them is amino acids, and you'll recognize some of these, hopefully, like dopamine, serotonin, and it also breaks it up into fats. So neurotransmitters that are created from amino acids versus neurotransmitters that are created from fats. There's also neurosteroids. So if you look into the chapter, there are more than just these, but these are some of the major ones, and these are kind of what we'll focus on. So here, we'll start with amino acids in just a minute, but we have single step. Basically, it only takes a single step to get from the amino acids to GABA and glutamate, versus for dopamine, and then norepinephrine and epinephrine, we have a multi-step process. So you take those amino acids and it's more steps to get to these ones. And then we'll see the synthesis of fats later on in the chapter. Hopefully you've read that already before watching the video. And then we'll just very briefly touch on neurosteroids. But again, this is not an extensive list of all neurotransmitters. Here's another image of neurotransmitters, some of their chemical makeups. And one thing that you'll notice here, this isn't a bad graphic, but when you see these functions, hopefully you're starting to realize as we're reading this that a lot of these neurotransmitters do many, many more than one or two or three things. All right, so here is how Wikipedia kind of breaks down the categories of neurotransmitter. But for our purpose, hopefully you can remember these two like major categories that the book uses primarily, which is the amino acid, the neurotransmitters created from amino acids, and the neurotransmitters created from fats. All right, so let's get into it. Some of the major principles of neurochemistry are that we're getting the basic structures or the basic building blocks for these neurotransmitters from our food. So that the food, the nutrients, the basic pieces that get built into a neurotransmitter have to get through the blood-brain barrier. So we have things like transporters that take those amino acids, those sugars, those fats or peptides, and they can bring them to cells. It's astrocytes often that are doing this process. And so these nutrients are basically just floating around in your blood after any given meal. Every cell in the body is needing these nutrients. So it's not just the neurons, it's not just the astrocytes, but every single cell needs some of these basics like sugars, amino acids, fats, and peptides. So with that, the blood-brain barrier has some specialized transporting mechanisms, and the fully formed neurotransmitters can't pass that blood-brain barrier. So pretty much they have to be made within the brain itself. All right. So enzymes do a lot of the job. They're converting those precursor nutrients into those neurotransmitters in this process. Those neurotransmitters, after they are created in the cell, are then transported into vesicles. Those vesicles then come down to the end of the synapse, and that neurotransmitter, when it's time to be used, is released into that synaptic cleft. And these neurotransmitters can then bind to the following cells. So they can cause those excitatory or inhibitory reactions and other secondary biochemical processes like being reuptaked by or being reuptaked into the previous cell or getting basically eaten up by those astrocytes and recycled in that way. So another thing here that is introduced in this chapter is these autoreceptors. So these autoreceptors are essentially detecting how much neurotransmitter actually went into the synaptic cleft to try and adjust itself. Say, did I put enough out there? Is there too little? Is there too much? Do I need to synthesize more? So do I need to take more of these precursors? Do I need to then make and obviously they're not actually thinking, right? But it's a metaphor. So these autoreceptors are detecting to know if the cell needs to make more of these neurotransmitters. So you can kind of think of these autoreceptors as a fuel pressure sensor in your car. So if you know much about cars, you've got the fuel tank and you've got your fuel lines and then you're squirting fuel into the engine, but you also have detectors that are saying, well, how much fuel am I actually spitting into that cylinder? And if you know how much fuel you're spitting in, if it's too little or too much, you can make adjustments, right? Your car's computer basically does this automatically, or a motorcycle's computer does this automatically, unless you've got something like a carburetor, but that's maybe going too far with this analogy. So essentially it's trying to detect, well, how much is getting spit out so it can know how much it should make. It can also send additional messages back up the axon. So that is a big part of learning and making these connections and building those networks in the brain. So here, the neurotransmitters are also terminated. So some of them are eaten up by enzymes, and so they're degraded, so they're not still there in the synapse. And this is happening pretty quickly. So let's watch a couple videos, a little bit of visual detail on how neurotransmitters can get into the synapse. And so we'll watch this first one and then we'll watch the second one. The audio should work just fine. You should be able to hear it as I'm watching it. On occasion, though, even though it's for educational purposes, sometimes a video will basically, I'll have to cut a video from my lecture slides, from my lecture videos. So if that happens, if I say, let's watch this video, and all of a sudden it's not actually that video, just go to the slides, watch it on your own. Sometimes it gets copyright struck and they'll just like mute it and then or they'll ask me to take it out. So this should be 2 videos in a moment, and then we'll come back here and talk about it. So this one first. This is a good example of how the neurotransmitters, especially those that are made in the cell body, like the ones that are made from fats, are transported down to the synapse. So not all are transported this way, but this should give you an example of some of the complex cellular mechanisms and biological mechanisms that are happening to move neurotransmitters where they need to be.
The human brain, one of our most complex organs, allows us to move, feel, think, and learn. Inside the brain, a dynamic network formed by billions of nerve cells processes information via electrical signals. Each neuron receives information from thousands of other neurons via multiple processes, called dendrites, and transmits information via a single axon. At specialized contact sites called synapses, electrical signals trigger the regulated release of neurotransmitters stored in synaptic vesicles. These chemical messengers bind and activate postsynaptic receptors, initiating a new electrical signal in the next cell. But how does a presynaptic cargo, for example, made in the cell body, end up at the right place within the cell? For this, neurons use active transport to sort specific synaptic proteins between the axon and dendrites. The microtubule cytoskeleton is important for this polarized cargo transport since it provides the roads for molecular motors. Microtubules built up from tubulin subunits are dynamic structures with a fast-growing and shrinking plus end and a more stable minus end. They have a unique organization in neurons. In the axon, microtubules have a uniform plus end out orientation, whereas in dendrites, their polarity is mixed. Two types of motor protein, kinesins and dynein, transport cargoes along microtubules.
So what we're looking at here are actually synaptic vesicles. So jumping to this, so inside those, we've got neurotransmitters. So some neurotransmitters are created closer to the actual synapse itself. Some are moved around through other processes in the brain between neurotransmitters and whatnot, which we'll see in a few minutes. But especially for the neurotransmitters that are created from fats, they're made in the cell body and transported all the way down these microtubules with these little motor proteins all the way down to the synapse.
Adapter proteins help to couple the correct cargo with the right motor protein. Kinesin and dynein move in opposite directions. Dynein moves towards the minus end, whereas most kinesins move towards the plus end. Therefore, the orientation of the microtubules helps to direct polarized transport. But many more levels of regulation exist to ensure proper sorting of cargoes. Various modifications of microtubules provide additional cues for motor proteins. Kinesins themselves are regulated by autoinhibition to prevent unnecessary activity. Attachment to a cargo or modifications of the motor protein releases autoinhibition and activates the kinesin. Multiple motor proteins can be attached to the same cargo, causing bidirectional movement. Inactivation or loss of a motor protein will cause the opposing motors to take the lead. Finally, reaching the right destination, the cargo is delivered, and in this case, released at the presynapse. Misregulation of neuronal trafficking caused by defects in the cytoskeleton organization, motor proteins, or regulators could all lead to abnormal neuronal transport, consequently resulting in synaptic dysfunction, neurodegeneration, and eventually brain diseases.
So pretty amazing to see that happening, to see, well, there's a couple ways that this can happen. These neurotransmitters can be synthesized up in the cell body or closer here to the actual synapse. But there's a lot of chemical processes that take it from that basic form of the amino acid, the sugar, the fats, the peptides, and then become the actual neurotransmitters themselves. So many, many complicated steps, and most of this is just happening just all the time. If you've got the kind of base nutrients, your body will just do it. So most new neurons in our brain produce two or sometimes three types of neurotransmitters, and they are released from those vesicles, and they can all be released at the same time. So if you've got, let's say, both GABA and glutamate, it might be releasing both of those at the same time. So let's talk about a few of these neurotransmitters that are made from amino acids. One of the biggest ones is glutamate. And like we mentioned before, it's called a simple amino acid neurotransmitter. And so it is only one step to get to this chemical, to get the chemical of glutamate. It's about 1,000 times more common than some of the multi-step neurotransmitters like dopamine. And glutamate specifically does excitation. It activates postsynaptic neurons. It tries to make them send a message. If you have too much activation from glutamate, well, you might have seizures or all the way to spastic paralysis. So doing too much so you're not actually able to move. GABA is another one that's very, very similar. It's also very, very common in the brain. So I think I've got a slide for it, not right here, but later on down. But I believe it's about 90% of neurons have are making glutamate and have it at the synapse. And then a closer to 30 or 40% of neurons have GABA as well. So still, that's way more common than other neurotransmitters like dopamine. And GABA is one that is inhibitory. It's kind of the opposite of the previous one, glutamate. So if you have GABA, it is trying to inhibit and slow down the activation or stop activation of the postsynaptic neuron or stop it from doing other things. So if you've got too much inhibition, you might have comas or other types of paralysis. So another one that we'll talk about pretty extensively here is dopamine. And so dopamine you get from the amino acid tyrosine, and then that gets converted to L-DOPA, and then it can get converted to dopamine. At that point, it then gets transported into vesicles, and then from that point, it can also be changed into norepinephrine, and then from norepinephrine, it can be changed to epinephrine. So it might make sense that you've got more dopamine than you have norepinephrine and epinephrine in your brain. Because you need that dopamine to then make that norepinephrine and the epinephrine. So you've got more dopamine, less norepinephrine, and then even less epinephrine. So one neat thing is that vitamin C, so one of those things that you're getting in a lot of foods that you eat, is something that sits with the dopamine or other neurotransmitters if it's using a vesicle, and keeps it healthy, like keeps it good, doesn't let it expire. So it improves its shelf life. So here is an example, and this image in your book is really to show like what is actually after you've used the dopamine, what breaks it up, and so that's what these things are over here. But I think it does a good job showing that this tyrosine becomes the L-DOPA, and so you can see this chemical process here, then becoming dopamine. That dopamine can become norepinephrine, that norepinephrine can become epinephrine. So the functions of dopamine and really all neurotransmitters depend on where they are in the brain. So what structure are they at? Are they at this structure, or this structure, or this structure, or this structure? That's really what leads to what does a neurotransmitter do. So the brain region defines the function. So neurotransmitters do a lot of complex things, lots of actions all over the brain, and it's different in different regions. So that is why it's incorrect, and the book really tries to emphasize this a couple times. It's incorrect or short-sighted to say something like dopamine is the pleasure chemical. Well, it's one of them if it's in the pleasure, one of those pleasure areas of the brain, but it's also helping with things like movement. So dopamine is one neurotransmitter made from amino acids. It's not as common as glutamate and GABA, but it does some functions all over the brain and kind of delivers itself through the brain in this pattern. So if you are in the substantia nigra, you go to the striatum from there, and if you are a dopamine, you are going to be working on motor function there. If you remember the parts of the brain, so we've got our sensory and motor areas right here. So that's why it's using motor function then. So some cell bodies in the ventral trigeminal area go to the limbic areas of the brain. And so that is where, over here, where we have a lot of controls of emotions with things like the amygdala, for example. And then if you're in the ventral trigeminal area and you go up to the prefrontal cortex, well, that's where consciousness is. And some of the pleasure pathways. There's a couple different pleasure pathways, but that's one of them. So depending on where that dopamine is, where it's activating, which presynaptic neuron to postsynaptic neuron it's activating. If it's in the limbic area, well, it's probably going to be working on emotions. If it's in the striatum, it's probably going to be doing motor function. If it's up here in the prefrontal cortex, it's probably going to be doing something with consciousness. And so this is an important key point that all neurotransmitters, their function depends on where they are. And each neurotransmitter does affect like certain receptors. As you read in the book, you see some have more receptors than others. So serotonin, for example, has very many receptors. But it depends where those receptors are in the brain. So that postsynaptic neuron that's getting that message from a dopamine or serotonin, well, what's that area of the brain do? What does that part of the network do? Defines, well, what is the function of this dopamine going to be? So this is an activity that we would do in class, so I'm going to skip it as we're going over the slides. But these are a couple really good videos on dopamine synthesis, and your book really dives in detail about the synthesis of dopamine, and I think that's a really good idea because it's one really good example. So the basic components of, well, what happens to take those precursors to make it into that dopamine? So that process and then getting to that synaptic, that synapse and then being used, that's very similar for a lot of neurotransmitters. So we'll go into a lot of detail here on dopamine, but then we'll kind of skim a lot of the other ones. So let's start with this first video. Again, if any videos don't work, go and like if I say, hey, this video, and then all of a sudden it jumps ahead, well, go and watch that video on your own.
While dopamine can be ingested in the diet, it cannot cross the blood-brain barrier, so the only dopamine that can be used in the brain is what is synthesized there. Dopamine can be synthesized from phenylalanine, tyrosine, or L-DOPA. Here you see the reactions where tyrosine is converted to L-DOPA with the enzyme tyrosine hydroxylase, and L-DOPA is converted into dopamine with the enzyme DOPA decarboxylase. Variations in the tyrosine hydroxylase gene, which involves a major step in dopamine synthesis, have been linked to neuroticism, anger issues, vulnerability, suicidal behavior, and some addiction problems.
So one of the main things with this one is just that visual example of the dopamine being changed by the enzymes around it. And so again, dopamine is a pretty simple one to make from amino acids. There are other neurotransmitters that are covered in the chapter that take many more steps than that to get to the final product, basically the final neurotransmitter that is used. As a reminder that dopamine can become norepinephrine and then epinephrine as well, with more interactions with other chemicals and enzymes in the brain. So let's watch video number 2.
L-DOPA is an amino acid involved in the synthesis of the neurotransmitter dopamine. When dopamine is produced in the brain, the amino acid tyrosine is first converted to L-DOPA, and then L-DOPA is converted into dopamine. While dopamine administered as a drug cannot pass the blood-brain barrier to enter the brain, L-DOPA can, and this characteristic has helped to make L-DOPA the most common treatment for Parkinson's disease.
So I'll let it go into a little bit of detail in a moment about how it's related to Parkinson's disease, but this is a key point to try and remember for all of these, is that amino acids, so neurotransmitters made from amino acids, go through this kind of process. You've got the pieces that come from your diet, your food, the things that you have in your bloodstream that are then changed to be a neurotransmitter with enzymes. Again, for most of these that are considered neurotransmitters, they do not pass the blood-brain barrier, so you've got to take the basic structural components and change them into the neurotransmitter that you can use inside the neurons. But let's see what this has to do with Parkinson's disease. We won't watch this whole video. The whole thing is pretty short and really good, but we'll just see the next little bit of how it's related to Parkinson's disease.
Parkinson's disease is characterized by the death of dopamine neurons in a region of the brainstem called the substantia nigra, and a subsequent deficiency of dopamine in a group of structures known as the basal ganglia. This dopamine deficiency is thought to contribute to the problems Parkinson's patients experience with movement. L-DOPA has a
All right, so they talk about how L-DOPA is used and given to patients, and it helps a lot of patients with Parkinson's disease for a long time because, one, it might be doing other chemical impacts in the brain, but also leading to more dopamine being produced. So hopefully you recognize substantia nigra from a few moments ago when we were talking about it in the slides as one of those places that you might have the dopamine created and then moved to, right? So right over here in the brainstem. All right, let's watch the next video.
Welcome to 2-Minute Neuroscience, where I explain neuroscience topics in 2 minutes or less. In this installment, I will discuss dopamine. Dopamine is a monoamine neurotransmitter, a term that refers to its chemical structure and the fact that it is derived from an amino acid. Dopamine is also a catecholamine, a term that also refers to its chemical structure and the fact that it contains a catechol nucleus. To synthesize dopamine, the amino acid tyrosine is converted to L-DOPA. Then L-DOPA is decarboxylated to form dopamine. There are several areas of the brain where dopamine neurons are concentrated. The largest are the substantia nigra and the ventral tegmental area in the midbrain. Other areas include the hypothalamus, olfactory bulb, and retina. There are several major dopamine pathways that carry dopamine from these areas of concentration to other parts of the brain. Some of the largest are the mesostriatal or nigrostriatal pathway, which stretches from the substantia nigra to the striatum, the mesolimbic pathway, which stretches from the ventral tegmental area to the nucleus accumbens and other limbic structures, and the mesocortical pathway, which stretches from the ventral tegmental area throughout the cerebral cortex. Dopamine acts at G-protein coupled receptors, and there are at least 5 subtypes of the dopamine receptor. Dopamine is removed from the synaptic cleft by a transporter protein called the dopamine transporter. Like any neurotransmitter, the functions of dopamine are complex and can't be fully explained with just a short summary. Dopamine is linked to movement due to disorders like Parkinson's disease that involve dopamine deficiencies. It is also often associated with the processing of rewarding experiences. However, dopamine also plays a role in many other functions.
So this is one of those points that we were talking about, is that depending on where that dopamine is in the brain is going to depend on what it's actually doing. So it's not just a pleasure chemical. It is activating those receptors at different parts of the brain. And depending on where they are, so where that presynaptic neuron sends it to that postsynaptic neuron, what network is that part of? That's going to depend on what it does. And so we see some commonalities of like some specific areas, whether it be emotion or cognition or things like that, or even pleasure. But it really depends on where it is in the brain. So hopefully these videos are helpful to get a little bit more of a visual example. The slides cover it, and we will talk about it in the slides as well. We'll go through it step by step. But hopefully getting that kind of visual example or seeing the interaction of the chemicals that become dopamine gives you a basis of an understanding of this is how a lot of neurotransmitters go through a process. And again, this is for amino acids or neurotransmitters made from amino acids. For neurotransmitters made from fats, similar but a little bit different. But some of those basic pieces, like the building blocks that it's getting from the blood, and those building blocks were things that we ate and things like that. There's a lot of similarities between all these neurotransmitters. So hopefully these big broad ideas of like what's going on with the synthesis of neurotransmitters, how do those neurotransmitters get moved to the synapse, and then they're released to the synapse. So as that neurotransmitter activates one of those ligand-gated channels, ion channels, you have sodium potassium ions moving and activating a new action potential. After that, you get it recycled. That neurotransmitter gets recycled, gets taken by astrocytes. Sometimes it gets received by the presynaptic neuron and detected or sends a message back up into the presynaptic neuron. But most often, a lot of it's getting other enzymes are breaking it down to be taken away. So hopefully those big, broad steps of this whole process are starting to make sense to you, that the repetition that we're getting from the reading, from the videos, and we'll go over it again in the slides right now. So, a little description again. You probably have a good connection with this since you read it in the book and you just saw these videos. But one of those first steps for dopamine specifically is tyrosine is transported out of the blood and into the neurons. So, again, we're taking some of those basic components out of the blood through that blood brain barrier, transporting that to the neurons themselves, where then we convert it into L-DOPA. So tyrosine hydroxylase converts that into L-DOPA. So that's what's happening here. Step 2. Then the L-DOPA can be turned into dopamine by dopa decarboxylase. So you've got all these steps. Then you take that dopamine, you put it in these vesicles. Again, in those vesicles, you have some vitamin C and you have this lipid layer. So that's how it can just become one with the lipid layer of the end of the cell. You can think of it, I think the book described it this way, that it's kind of like two bubbles connecting and sharing one set of those lipids. But so that's step 4. You transport that into the vesicles. Then you could, if you wanted to, if your body's doing that in these neurons, is convert that to norepinephrine. So you've got that dopamine, you can turn it into norepinephrine with that dopamine hydroxylase, or you can then take that norepinephrine and convert it to epinephrine by this PNMT in those vesicles. So that's how you get the dopamine, you can get norepinephrine, and you can get epinephrine, all from those same basic components that you're getting from the blood. And how did it get in the blood? Well, you ate stuff that broke down in your digestive system to give you those basic components that become this neurotransmitter. So, this is a really good video, a lot more detail than you need to know in this class. But if you want to know more about the chemistry of how we get from that tyrosine to the dopamine, I definitely recommend watching this. We're not going to watch it now in the recording, but if you want to get more in depth into the chemistry of that, this is a really good one for that. It's also about 18 minutes long, so that's another reason we're not watching it in class. All right, so now you've got the dopamine. What are you going to do with it? Well, when you get the proper signal to your neuron and you're the presynaptic neuron, you're going to spit out some of that dopamine into the synaptic cleft. And we've got these D1-like receptors and D2-like receptors. So we've got a couple different types of receptors. And one of those videos mentioned there's even more than these. But this is a couple different things that it can do. So you can either activate or inhibit this so that you are getting a different response in the postsynaptic cell. So essentially, the dopamine, depending on the receptor, is going to lead to activation or inhibition, or excitation or inhibition. So now after you've used that dopamine, either excited or inhibited, the postsynaptic neuron, well, then you've got to be broken down by MAO or COMT, and you can then, you're getting rid of that very, very quickly after you have sent it into the synaptic cleft. So again, a really important thing to get across here is that what the dopamine is doing depends on where it's located. Again, if you are located in the prefrontal cortex, you're probably going to be more involved with things like consciousness and activations of some of those networks, versus if you're in the limbic system, probably more about emotion. Same with norepinephrine. No matter what kind of neurotransmitter we are talking about, it depends where it is. Where it is going to let you know, well, what's it going to be doing there. So that is all on dopamine. Hopefully you get a good sense of the pattern of what happened with dopamine and with those videos. Hopefully that helps because we're not going to go into as much detail on most of the rest of the neurotransmitters. We'll hit a couple specific interesting facts, but hopefully that kind of pattern of taking the precursors, creating the neurotransmitter, getting it to where it goes, and then recycling it and it activating specific receptor sites. Hopefully all of that is starting to make sense. You're seeing these patterns with these different neurotransmitters. So the next one we'll go into in not as much detail, but the next one we'll talk about is serotonin. Now going into serotonin, a lot of it is pretty similar to what we just talked about with dopamine. So hopefully with that example of dopamine, you've got a better understanding of the basic processes that we're talking about here. So for serotonin, there is some unique facts that I want to get across. Your book did talk about this, but it is a ancient signaling molecule. It's been around for many, many millennia. So essentially the entire time that we humans have been evolving, serotonin has been used in signaling at some point. So even insects, single-celled organisms, bananas. So honestly, I need to look that up in a little bit more detail because that's really interesting. And humans, we all synthesize it. We create serotonin. So with that, because it's been around for so long, there are a lot of receptors. So we looked at basically two receptors for dopamine, one that basically leads to excitation, one that basically leads to inhibition, but there are other dopamine receptors, but basically about five versus for serotonin, there are a lot more. And so with this, there are different ion channels, there are different chemicals that it's activating at the neural level. So you've got a neuron that has a serotonin receptor, right? So depending on what that receptor is, depends on exactly what it's going to do at that receptor site. Is it going to let some sodium or potassium pass, right? Or is it going to activate other of the channels on that membrane? With this being said, this is another reason that we see serotonin all across the body, including in the gut, and it is a part of signaling in a lot of areas. So the processes of what is serotonin doing, again, depends on where is it sending the signal from. Is it sending the signal from this area versus this area is going to depend on what it leads to. With that being said, most serotonin is actually retransported back into the presynaptic terminal and recycled that way. So it's reuptaked. So serotonin in the synaptic cleft, anything that is there, is broken down by MAO and aldehyde dehydrogenase. Oh my goodness. You read these words and it makes perfect sense as you read it. It's like, yeah, I remember that's related to this. But then you try to say them, and sometimes it's been a while since you've heard it said. So, and then some people say things differently as well. So, like I mentioned for serotonin, it's been around a very long time. Serotonin has basically been with us the entire time we've evolved into the uh, humans that we are now. So 90% of serotonin that we have in our body is actually in our gut. About 8% is in blood, so that's like platelets, mast cells. 2% is in the pineal gland, and then about a fifth of a percent or a half, about a half of a percent is in the brainstem, but it does project widely throughout the brain. There's just a lot more of it in other areas of our body, like our gut. So with this, it is helping with a lot of different things across the body, a lot of messaging between organs, keeping things at homeostasis, trying to keep our body functioning as it has evolved to do. Now, we will jump ahead to other types of neurotransmitters from amino acids. So that's kind of the point with a lot of these neurotransmitters. Serotonin is distinct and different from dopamine in some really interesting ways. This is one of those ways that it has a lot more receptors, that a lot of it's in the gut, that it's used for messaging across other areas of the body, but it is used for messaging in the brain between pre and postsynaptic neurons. So, while we're not going to dive into as much detail as the book did about how it's synthesized and things like that, that's kind of the idea for the rest of these is highlight some of the distinct differences, similarities about the different neurotransmitters. So neuropeptides are a little bit more complicated. They're a little bit more complicated than the dopamine and the ones that take only just a step. So things like GABA and glutamate. So with this, they can be just 4 or more amino acids together, or they can be hundreds of amino acids in really complex three-dimensional structures. And these often are releasing with other neurotransmitters. Remember, some of the most common in the brain are glutamate and GABA. So with these neuropeptides, these specific types of neurotransmitters take a lot longer to synthesize. They can take 2 to 4 weeks to synthesize because for this process to create some of these neuropeptides, you have to read the DNA, produce the mRNA, and then translate that into a long string of amino acids. And so we've seen that in some videos in previous classes of basically copying the DNA to the the mRNA, and then taking the amino acid and connecting it to that. Sometimes that is a neuropeptide and a neurotransmitter. So these are an example of one of the types of neurotransmitters that uses those microtubules that we looked at the video of a little bit ago. So here is the picture in the correct orientation. Essentially, so here in the nucleus of the cell, you're reading the DNA, you're producing the mRNA, and then you translate that to a long string of amino acids. It might be just 4, it might be 100 amino acids put together. Then you put that into a vesicle, transport that down the axon. Again, you're using those motor proteins, you're getting it here to the end, and you're using it in action potential. But again, this is a lot more complicated and longer process than some of the other ones that are synthesized closer to the actual the synapse itself. One example of a neuropeptide is endorphins, and there are a lot of classes of endorphins based on the chemical structure. Hopefully, as you've seen with biology in general, chemistry in general, we try to categorize them, to categorize things, put them in literally little boxes that can kind of match up so we can kind of understand what things are related to one another. But there are a lot of different of these neuropeptides. So some things that they might do is regulate endocrine production, immune cell production. They might be related to pleasure and euphoria feelings. And they modulate pain. So your book dives into a little bit more detail about pain. We will come back to this as we get to week 7 when we're talking about the perception of pain as well, and how these play a big part in sending those messages. Now, jumping forward to another specific one from amino acids, oxytocin. You've probably heard about this a lot. So this is a chemical that people talk about as the love chemical, and there's a couple specific interesting features about it. So it is being expressed, it's being synthesized in this specific area of the brain, and it's released into the bloodstream here around the pituitary gland. It's also released throughout the limbic system and into the cortex, but there's a big process of releasing it here into the bloodstream, so that that oxytocin can message with other organs as well as neurotransmitters for pre and postsynaptic neurons. With this being said, some of the functions that we see from oxytocin, social behavior, reducing social anxiety, mother-child bonding, it's pretty impressive how we see more oxytocin being delivered in parents with children, especially with things like skin to skin contact, even immediately after birth. So, being able to measure those differences in that cortex and the limbic system, seeing that a person who has just given birth has so much more oxytocin that is being released. So, it's a big part of a lot of processes that help us make connections as humans, which is a really important thing for us as the social creatures that we are. All right, we'll jump back to some of the simpler ones that are made from amino acid, both glutamate and GABA. So with this, glutamate is primarily an excitation, and it's the most common excitation neurotransmitter in your brain. Again, it is much more simple to synthesize than some of the other ones that we've just been talking about. And then GABA as well is much more simple to synthesize. So both glutamate and GABA are the most common neurotransmitters in the brain. 90% of neurotransmitters use glutamate, and then 30 to 40% of neurons use GABA. So with that, again, glutamate is the excitation one, GABA is the inhibition one. Basically, they're kind of balancing each other out. They're firing competitively. Any given neuron might send both of them into the synaptic cleft in a specific combination of both excitation and inhibition. So with GABA specifically, so the one that's a little less common and the one that is doing inhibition, neurons, as we learned in chapter 2, are leaking those potassium ions. So because of that leak, it is possible for just random sparks of network activation. GABA helps stop that full network activation. GABA is there to say, well, I'm not getting other signals around, so I can stop this random activation. So that being said, though, it is an important process in inhibiting those, in stopping those activations, which is very important for many, many things that we do in our brain and helping us intentionally do things, which then we'll jump back to glutamate, which again is that excitation hormone or excitation neurotransmitter that's really important in 90% of cells. They're delivering it to activate the next one. So one of the main things with glutamate is that it's important for learning, and we'll come back to glutamate as we get into week probably 8 or 9 as we're talking about learning and memory. So one of the big things is that as we learn, as we are trying to make connections, as we are building those networks, we need to be able to activate and let postsynaptic neurons know that I should activate now. One of the ways to do this is the glutamate, especially because it is so prevalent in the brain. So let's watch this video about glutamate and talk about it in a little bit of specificity of how is glutamate leading to those networks being created. The presynaptic neuron talks to the postsynaptic neuron the first time. What happens the second time or the third time or the fourth time? Once this, how do you get to a solid highway versus a little path of a connection?
Welcome to 2 Minute Neuroscience, where I explain neuroscience topics in 2 minutes or less. In this installment, I will discuss long-term potentiation, or LTP. LTP is a process by which synaptic connections between neurons become stronger with frequent activation. LTP is thought to be a way in which the brain changes in response to experience, and thus may be a mechanism underlying learning and memory. There are a number of ways in which LTP can occur. The best-known mechanism involves a glutamate receptor known as the NMDA receptor. In NMDA receptor-dependent LTP, glutamate release first activates a subtype of glutamate receptor known as the AMPA receptor. NMDA receptors are found nearby these AMPA receptors, but are not activated by low levels of glutamate release because the ion channel of an NMDA receptor is blocked by a magnesium ion. If frequent action potentials cause greater stimulation of AMPA receptors, however, this will cause the postsynaptic neuron to depolarize, which eventually causes the voltage-dependent magnesium blockage of the NMDA receptor to be removed, allowing calcium ions to flow in through the NMDA receptor. This influx of calcium initiates cellular mechanisms that cause more AMPA receptors to be inserted into the neuron's membrane. The new AMPA receptors are also more responsive to glutamate and allow more positively charged ions to enter the cell when activated. Now the postsynaptic cell is more sensitive to glutamate because it has more receptors to respond to it. Additionally, there are thought to be signals that travel back across the synapse to stimulate greater levels of glutamate release. All of this makes the synapse stronger and more likely to be activated in the future. This process is also associated with changes in gene transcription in the neuron, which can lead to the production of new receptors or modifications to the structure of the cell. These changes seem to be important for making the increased responsiveness of LTP long-lasting.
So jumping back to this specific part of the video, when we're talking about glutamate and its excitation properties, whether it's memories, whether it's habits, whether it's something that you've learned to do. So one example that we've given in class is maybe learning to play guitar or piano or maybe knitting, right? So you've learned to do things, or learning a new language, whatever it is, whatever memory it is, you are using this basic process. That glutamate, again, is in 90% of your neurons, and so it's very much active all around different areas of your brain. And it's one of the keys to building that strong network. So as the glutamate is activating these AMPA receptors, it can activate this receptor, which its name just left me, the NMDA receptor. And when that happens, the cell will begin, again, this is the postsynaptic neuron. This is the membrane of the postsynaptic neuron. It will synthesize and create more of these receptors, the AMPA receptors, which means if you have the same amount of glutamate spit out into that synaptic cleft, if you just had these two before, well, this one would activate, this one maybe would activate, right? But now you've got four here. Well, you're going to get a lot more activation, a lot more likelihood to get that time series or the amount series to start an action potential. So essentially, you now have more receptors for the glutamate. So that habit, remembering how to say a word in a different language, remembering how to hold your hand in a specific way for a chord on a guitar, how to hold your hand in a specific way for a chord on the piano, how to hold your body in a specific way and your hands in a specific way for knitting. Think of another example that's not so hand focused. Driving. When you started driving, some of those processes or figuring out where something is in the car was a little hard, right? If you drive a friend's car or rent a car, the buttons might be a slightly different place. The turn signal might be a slightly different place. The button for the volume or the knob for the volume, right? So these might be in a little bit different places. But over time, it becomes more second nature to activate the turn signal or to turn up or turn down the volume, right? And part of what is happening there is this process of adding these AMPA receptors makes it so that network for, oh, instead of using this network, use this network is easier to activate. And that's part of the same process of forgetting as well, is losing some of these receptors because there's not being used. Again, the brain, the cells themselves want to be efficient. All right, so that is one of the reasons that glutamate is so important in learning and memory formation. And we'll come back to it again as we talk about memory specifically, but it's very similar. And then another couple points is, as well as that presynaptic neuron can produce more glutamate, get that signal back and then know, hey, I should be producing more of this. It can also create more axon and dendrite branches as well. So that means there could be even more signaling to those postsynaptic neurons that are part of that network. All right, so this is an activity that we're going to do in class. If we're not in class, then that is too bad. So I'll skip it in the review of the slides. And let's jump to one of the little bit more complicated steps is making neurotransmitters from fats and lipids in the body. So these are called endocannabinoids. Endocannabinoids. So some you might see some commonalities for things like cannabis, for example. The book actually goes into that example. Your weekly activity goes into that as an example. But there are different endocannabinoids that are neurotransmitters that send that message that are much less common than things like obviously glutamate or GABA, but even dopamine. But there's at least seven. Some of the most well known are anandamide and 2-AG, and these are not stored in vesicles. They're actually synthesized in the postsynaptic cell. So that's a unique thing about some of these neurotransmitters made from fats. So one of the interesting things about these is retrograde neurotransmission. So they're released by the receptor cell. So when they're getting a signal as they're having the neurotransmitters activating the voltage gated cells, they can send these neurotransmitters made from fats into the cell to signal that it needs to do something slightly different. So the neurotransmitter releases and activates the postsynaptic cell. The postsynaptic cell activation activates both the DLG alpha and the PLD to synthesize these neurotransmitters made from fats. Then it diffuses across the cell membrane to the synaptic cleft to activate receptors on the presynaptic neuron. And the basic idea here is it is inhibiting the release of a variety of neurotransmitters. It's altering the function of the potassium voltage gauge, the potassium calcium voltage gauge channels, and it can be related to motor suppression, memory impairment, appetite stimulation, reduced stress, immune modulation. There's a lot of things that it can do. So this is another example of the message coming back from the postsynaptic neuron. So we do have those sensors on the presynaptic neuron to see how much neurotransmitter went in, but we also have these endocannabinoids that can send the message back that way as well and lead to inhibition or altering or modulation of the function of those cells. So one example of this is acetylcholine. I tried to find so many videos about acetylcholine for this lecture, and it's so funny how many different pronunciations of acetylcholine I heard. But again, for any of these, acetylcholine, GABA, dopamine, remember the function depends where in the brain it's being used. So some things that we see for acetylcholine is it's very related to learning and memory. So hippocampus, hopefully that is getting familiar to you. Amygdala, hopefully when you hear amygdala, you think of things like emotions. When you hear things like the frontal cortex, you think of things like attention. And then when you hear something like the cerebellum or the motor areas of the brain, you think of movement. All right, with this being said, this is related to Alzheimer's disease. Again, we will get into specific kind of diseases and disorders later on in the class, but this is a really good example of a specific disease that's very related to acetylcholine. So Alzheimer's is a degenerative brain disorder. Basically, you can see the healthy brain here on the left, a diseased brain on the right. And one of the big things that's happening here is the death of neurons. And there can be quite a few things that can lead to that death and trigger this, which we'll get to as we go. But a big part of it is a degeneration of those acetylcholine producing neurons. And because it is part of things like memory in the hippocampus, it can lead to memory problems. In other areas of the cortex, it can lead to attention problems. And so that's why we see some of the symptoms that we see, because some of these neurons that produce the acetylcholine are dying. So you can use inhibitors to prevent that from happening. You can do other things in addition to having those inhibitors. Learning more, trying to build the plasticity can reduce the damage that Alzheimer's does over time. But at this point, we don't have something to completely stop the death of those neurons. So it's kind of like these treatments are slowing the progression of the disease over time. So we are at one of the last points in the lecture, the neurosteroids. And the book barely even covers these, but they are an important part of a lot of processes in our brain and across our bodies. If you are familiar with neurosteroids or steroids in general, you might recognize the name or the steroid progesterone. Also, another example is pregnenolone sulfate. So these modulate brain activity, can make it more or less active. They interact with neural membranes and ion channels, making them more or less likely to fire. They're also anticonvulsants in other areas of the body as well, not just the brain. Progesterone is one that's important in, say, a lot of hormonal systems and giving birth. So there are, and this is actually a really great article. If you want to know more about neurosteroids across the lifespan, all of the different systems that they are involved in, this is a fantastic article that was published pretty recently as of my recording this video anyway. And they also interact with glutamate and GABA receptors to modulate, to make them more or less activated, excited, or inhibited. So they're a big part of that puzzle. Hopefully, as we've gone through this chapter, you see that there are a lot of pieces to that puzzle. It's not just the simple dopamine or serotonin make pleasure or happiness, right? Depends on what area of the brain. There's a whole process of synthesizing each of these neurotransmitters. There's a whole process to how to get it to that end of that synapse and then into the synaptic cleft. Then what receptors are receiving it? What exactly do those receptors do? Does it send a message back up to the presynaptic neuron? How is it recycled or reuptaked or deleted or taken away by enzymes or astrocytes? So hopefully these examples, as we've gone through, help you see some of those things and also realize how complex the whole system is beyond just that dopamine leads to pleasure or something like that. Hopefully you've got a lot clearer idea about that, because it's important as we go forward and see all the really interesting specific things that it leads to, whether it's with our learning, as we just barely touched on today, if it's with a disorder that we barely touched on today. These are important pieces all throughout the brain and the rest of the body as well, in the different combinations that lead to the behaviors, the activations, the networks that we have to lead to who we are and how we do things. So this is a funny song about a lot of things from both chapter 3 and chapter 2. I won't watch it in this video, but I definitely recommend watching this one. It's pretty silly, but if you're trying to remember some of these terms and you're getting things confused, putting things to song or rhyme is a really good tool for human brains to make those connections. This is another good review for a lot of the neurotransmitters in the book. It's basically the full version of those 2-minute neuroscience videos that we watched for like dopamine. So it goes over all of them in order. So it's another good example if you want a review. But as for that, this is the end of the video lecture for chapter 3. Thank you very much for being here. Hopefully you've got a little clearer picture of how these neurotransmitters are working around your brain. And hopefully you can remember right now for a quick pop quiz of which one is the most common. So if not, maybe rewind and review that section, but it's glutamate and then GABA follows. All right, I'll see you in class or see you next time in the video.